Hello, my name is Siobhan Green. It is week 3 microbiology. I am doing experiment 2 of week 3's. So it's bacterial morphology and staining techniques. I'm going to go ahead and begin with exercise 1. So the first step it says is to label my slide, which is with one direct in. We want it to make sure it dries because I then have to flip over the slide and make a dime-sized circle in the center of the slide. So as you can see there, I have made my circle and I have labeled it. The next step is to vigorously rub my gums and teeth with my swab. And then we want to smear it Okay, so it says let it completely dry, which I have done. It says to light my tea candle, which I have right here, as you can see. Pass the slide over it 5 times. Okay, and now to let the slide dry for 10 minutes. Okay, so I have let it cool down for 10 minutes. I am going to place it in my staining tray. I am going to take my crystal violet gram stain and add 1 to 2 drops. Okay, so I have added 2 drops, and it says to let its stain sit on there for 60 seconds and then to wash it off with water. So I'm going to do that. So I have removed the stain from my slide, rinsed it off, blotted it, and now I'm going to stick it into my microscope and look at it in 3 different powers, the scanning low and high, and then take my photo and insert it. I am now doing my second slide, where I'm going to take my Congo red and put a drop onto it. Okay, and so it tells me to take my second swab and do the same thing with my teeth and gums. And now to agitate the stain to collect the cells from my mouth. And I'm now smearing my stain across my slide. And I'm going to let it dry. I am now on exercise 2. I am doing differential stains, so I have labeled my slide E. coli and I have my dime size circle in the middle. So it says remove this, lighting my tea candle, I've sterilized my loop, and it wants me to remove the E. coli lid and pass through the tea light. Okay. And now it's telling me to take my tube or my loop and insert it into the broth. And transfer my liquid onto my slide here. Okay, so I'm now gonna take my slide and put it over the heat, doing a heat fixing sample. And then I'm gonna repeat with my 2 other slides that I have prepared also. So that was the E. coli. Okay, so now I'm going to add 2 drops of crystal violet, and I have my E. coli in there right now. Okay, and I'm going to let that sit on there for 60 seconds and then wash that off. Now I'm going to add 2 to 3 drops of the PPB iodine to the same E. coli where I did my violet. Going to wait 60 seconds. Now 2 to 3 drops of my decolorizer. And I'm going to wait 30 seconds. And now my safranin, 2 to 3 drops. I'm going to wait 30 seconds for this one. I will rinse it off, and then I will lay it on the towel to dry and do my yeast and my epidermidis. So I have prepared all my slides with the stains, the E. coli, the yeast, and the S. epidermidis. My E. coli is dry, so I'm going to go ahead and put it in my microscope and look at it through my powers, take my pictures, and then insert into my lab assistant answering the questions and those type of things. And I will do the same things with the other 2. And that will be the end of the experiment. Thank you so much.